Hello, so the second part of cells then is all about microscopes. So microscopes are really handy obviously because they allow us to see cells which are very, very small. You might even say that they're microscopic. Now, we've got several parts to microscopes which are important. This is a typical light microscope because it uses light to view the image. Later on, we're going to be looking at an electron microscope, which is slightly different and more complex. Now, the main parts that you need to know about is the eyepiece lens, which is basically just a little lens which goes close to your eye, which magnifies the image. It usually has a magnification of ×10. Then we've got an objective lens, which goes close to the object and magnifies the object that you're looking at. You can have lots of different ones. You can have ×4, ×10, or even ×100. Then we've got clips and a stage. Now this is where you put the little glass slide with your sample on, and you just use the clips to hold it in place. The stage can be moved up or down, closer or further away from the objective lens, and that just helps you focus the image. We've got a coarse focusing knob and a fine focusing knob, and both of those move the stage to help focus the image. We've then got a light source which sends light upwards through the specimen here into the objective lens, up through the eyepiece lens and into your eye. So as I say, here's another slightly different microscope. There's two lenses, the eyepiece lens, which is what you look through, goes close to your eye, and the objective lens to make the object appear bigger. We then have a focusing wheel, which moves the stage up and down to make the image clear. And this is just a little function of each part. And this part just shows you how you actually use a microscope to focus an image. So you always start with a low power objective lens. Now that usually has a magnification of ×4 or ×10. You make sure that your image or the sample is in the center of the circle of light. If it's wide, it allows you to see more of the cells on the slide, and it makes it easier to find what you're looking for and focus a sharp image. After you observe the cells at low power, you may want to see in more detail. So to do this, you move the cells to the center of the field of view and then use a higher power objective lens. So the steps to focusing an image: you place the slide on stage. You start with low power, and that allows you to find the cells, because that can be a bit tricky, and to focus the image. You then use the focusing knobs to move the stage and focus the image. Then you change objective lens to a higher power, and you would do that to see in more detail. Now, one wee practical thing about this: when you are focusing an image, you place the slide on the stage and start with low power. Now, you then use the knobs to move the stage. It's important to say here that you want to move the stage downwards first of all. Now, the reason why you would do that is that you don't want to damage the lenses or the microscope slide. Now, what I mean by that is this stage, as I said, can move up or down. So if you start with it low and move it up, as you're trying to focus it, you're going to be looking down here. So you won't actually see how close this stage gets to the lenses. So what you want to do is move the stage up here as close to the lens as possible and then focus it by moving it downwards. So that means you're in no danger of damaging the microscope. A common practical then at GCSE would be making slides of onion cells and making slides of cheek cells. So if we have a look at each one, what you do is use forceps to peel a thin transparent layer of epidermis from the inside of a small section of an onion bulb. Now the reason why you want to make it thin is so light can pass through the cells. Because if light doesn't pass through it, we can't see it. Okay, so what I mean by that is we're going to put our little onion slides on a microscope slide there. For us to be able to see it, the light has to travel from there through the sample, up through the lens, and up into our eye. So if it's not thin, the light can't pass through. So we get a thin layer of cells from an onion. You place it onto the microscope slide, which is the long rectangular part. You add iodine, and the chemical stains the colour, making certain parts such as the nucleus more obvious. And then you cover with a cover slip. And you just have to make sure that if you lower one end first, it helps prevent trapping air bubbles. Now, the next thing you could do is you could prepare a slide of cheek epithelial cells. So this time what you do is you rub a cotton bud over the inside of your cheek, or you swab the inside of your cheek. You then smear the cotton bud onto a clean microscope slide. This time you add methylene blue and cover it with a cover slip. Now, in each one, we use stains like iodine or methylene blue because it allows the structures to be seen more clearly or more easily. If we didn't stain these cells, which is a typical cheek cell, you wouldn't be able to see it as easily. Now let's summarize that. So if you've got to make a microscope slide, here's what you do. You get the cells, you place on microscope slide, use a stain, and then you cover with a cover slip. Now, if you're looking at onion, you peel a thin layer. So I'm going to write onion in red. So you peel a thin layer, you place on a microscope slide flat, and then you stain with the onion its iodine. If you were using cheek cells, you swab your cheek, place on a microscope slide, and then for cheek cells, it is methylene blue, and you cover with a cover slip. So red writing is to do with onions, green writing is to do with cheeks. So you've done these experiments. What do you see? For the onion cells, this is the sort of image that you would see. And the things that we can make out are the nucleus, which is here, here, and here. Each one of these little things is an individual onion cell. So there's one, there's another one. The cell wall is around, cell wall is around the outside. The cell membrane is really, really thin. So we basically just label that as the inside of the cell wall. And then the area inside is just a vacuole. So what we can make out is a nucleus, which is here, cell membrane, which is just inside the cell wall, cytoplasm and vacuole, and cell wall. But they don't have chloroplasts. And that's because chloroplasts are used for photosynthesis, and onions don't photosynthesize because they grow underground. Now, cheek cells here, this is what you would see. So they appear blue because we've stained them with methylene blue. The only things we can really make out here are the cell membrane, which is around the outside, the cytoplasm, and the nucleus. So, way less to see in the cheek cells. Now, we just said that we used the stain so that we could see them more clearly. So the next little part then is an important skill, recording what you see in the microscope. Now, usually you have to draw these. So when you're making a drawing of what you can see from a microscope, you need to make sure that they're made in pencil, with firm continuous lines, not sketchy with gaps, large and in the same proportions of the cells observed, and you label them using separate lines. Now, here we've got a little cheek cell. I'm going to try and draw these cells in here following these rules. So we can clearly see that we've got two cheek cells. Now, a little tip is draw an outline of them on your photograph, and that'll show you what sort of shape you need to draw. So this one we'll start off with first. It needs to be smooth, continuous lines, not sketchy. So this would be an example of sketchy lines. They need to be continuous. And then we need to draw the little nucleus in. And that's roughly what you should draw. There's nothing fancy about it. You shouldn't spend too long on it and overthink it. Just make sure that you sketch over your photograph and then you draw exactly what you want to draw. Now, it should be in the same proportions, which means that it should be the same size. And most of all, the one word that you're trying to think about here is your drawing needs to be representative. So what you're seeing in this photograph, that's what your drawing needs to look like. It needs to be a good representation of your photograph. A bad representation would be me just drawing two cheek cells like that and then labeling. Which brings us on to the last bit. Let's label it. So we have cell membrane, nucleus, and cytoplasm. And that's how you draw. Now, measuring cells then. So before we get into measuring cells, it's important to understand the units of measurement that we would use for them. Now, we're not going to use centimeters because they're too big. We rarely use millimeters because again, they're too big. So instead what we use is this little which is known as micrometers or micrometers. Now, it's important to remember that there is 1000 micrometers in 1 millimeter. This little image just shows you how you would go about converting images. So as part of questions, it's pretty standard that you would have to measure something using a ruler. You might measure it in millimeters and then have to convert it to micrometers. So if you have to do that, you take your value in millimeters and multiply it by 1000. So let's say I measure something, a little scale bar underneath the photograph, and I measure that, and I work out that it's 70 millimeters. Part of the question might be that I convert that into micrometers, so I'd have to times that by 1000. So it would be 70,000 micrometers. Now, I'll show you examples of that later on, but this is the important bit that we are taking away from this part. 1000 micrometers is equal to 1 millimeter. Now, typically whenever you look at cells under a microscope, you might want to measure them. And that's just to give you an idea about what size the cells are. Now, obviously they're very, very tiny, so we can use a stage micrometer to try and work out the size of them. This would be very, very small. In real life, it's 1 millimeter in length, so it's extremely small and difficult for us to see. So what you do is you put it on your microscope slide, and when you look down your microscope, you see the field of view, and this is what the stage micrometer looks like. So say, for example, we had a cell and we were looking at it under the microscope, we could actually measure its length, and we could tell that in real life this cell was approximately 50 micrometers or 500 micrometers, which would be a pretty big cell. So the important thing here is that to measure cells, use a stage micrometer. Now, let's look at a few little conversions. So remember, there is 1000 micrometers in 1 millimeter. So if we've measured a photograph and it's 22 millimeters, how many micrometers are in that? Multiply it by 1000 is 22,000 micrometers. In a drawing, an onion cell measures 37.5 millimeters. Times it by 1000, it should be that. Now, just for a bit more practice on that, pause the video here and try and convert these units into whatever I've told you. So, for example, 1000 micrometers into 1 millimeters would be, bosh, 1 millimeter. So pause it here, have a go. And here's the answers. Now, in exams then, sometimes you'll have to use this little equation to calculate different things. Sometimes you'll have to calculate the actual length of a cell. So we know that cells are tiny, so we might have to calculate its actual length. Now that'll be a very, very small number, and we're usually talking micrometers there. Or it might be the magnification, which is how many times the image has been zoomed in on. It's not that often that you'll have to calculate the length of the image, but what you might have to do for that part is use a ruler. So let's look at what each part means. So examination questions ask you to calculate the actual length, which is pretty much the length of the cell in real life, or the magnification of a cell, which is how many times it has been enlarged. How the size of the cell or structure as seen on the diagram or seen on the drawing or photograph is called an image. So what you actually see when you look into a microscope is an image. A drawing or photograph of cells under the microscope has been magnified. Magnification is the number of times the length of the image is larger than the actual length of the cell. Now, if we look at this, this is the little equation you can use. Length of the image is that. Now, sometimes you'll be given two of them and have to work out the third one. So, if, for example, you had to work out the actual length, you just cover it up and you would do the image length divided by the magnification. So let's have a look. Now there's different ways you can do this. You can calculate magnification using a scale bar. So sometimes magnification can be represented by a line called a scale bar on or near a photograph or drawing. The length of the scale bar can be measured in millimeters, converted to micrometers, and divided by the actual length to calculate magnification. So, if for example you had to calculate the magnification, you would take the length of the image and divide it by the actual length. So here's an example. This is a typical little scale bar. So scale bars are usually seen at the bottom of microscope images. Now, you take the image length of the scale bar and it's 20 millimeters. Now, that had to be measured using a ruler. So you would have to physically take a ruler. Oops, pardon me. You would have to physically take a ruler and measure its length. The next thing you do is convert it to micrometers by multiplying by 1,000. So as long as you've got it in millimeters, you multiply by 1,000 and that gives you micrometers. The actual length of the scale bar is 1,000 micrometers, and that comes from there. So that's what size this would actually be in a microscope image. You then do magnification equals the image length divided by the actual length, which equals 20,000 divided by 100, which equals 200. So that means that that scale bar has been magnified 200 times. Now, let me show you what it would look like in an actual exam question. So here is some red blood cells. Use the scale bar, calculate the magnification of the image below. So, magnification equals the image length divided by actual length. So, what we would need to do is work out the image length, and to do that we need a ruler and it needs to be in millimeters. So you would take your ruler. Now obviously I'm just showing you what this is here, and I can't really show you exactly what length it is because this isn't a ruler in millimeters, it's just because it's on the computer. So in real life, on a piece of paper, this image, the length of the scale bar is 53 millimeters. You then need to convert that to micrometers, which is 53,000 micrometers. The actual length is usually written on the scale bar, and it is 50 micrometers. So what you do to work out the magnification, you take the image length, which is 53,000, divided by 50. Now let's just keep track of where we're getting all these values from. So there's our image length, and there is our actual length. So that means our magnification is times 1060. Now what that actually means is this image is 1060 times larger than it is in real life. Now of course you can rearrange that and calculate the actual length of a cell. So in this case we had IAM. So if we want to calculate the actual length, we need to take the image length and divide it by magnification. So let's have a look at it. Magnification of a photograph taken under the microscope is times 250. A student measured a cell on the photograph to be 15 millimeters. What is the actual size of the cell? So actual size of the cell equals image length divided by magnification. So that means we need to take the image length, which is 15 millimeters, convert that to micrometers, which was times it by 1,000. Magnification is 250, so we simply need to do 15,000 divided by 250, which equals 60 micrometers. Looks so simple, but that is the part which catches people out. That and not arranging the equation properly. So anytime you see millimeters, you need to remember to convert to micrometers. which is timesing by 1,000. The last question here then is to calculate the actual length of a cell. So what size is it in real life? So to calculate the actual length, you need to take the image length and divide it by the magnification. So you get your ruler out, you measure the image length using the scale bar. When you measure it, it's 66 millimeters. Convert it to micrometers, which is 66,000. The magnification is given to you here. It's 350. So really simply, you just do 66,000 divided by 350, which tells you that the cell's actual length in real life is 189 micrometers. Now, if you're doing this and you're thinking, I don't know if this is right, remember, cells are very small. 189 micrometers is really small. If you rounded that up, it's 200 micrometers, which is basically 0.2 of a millimeter. Does that sound right? Yes, because cells are tiny. So the last little section of this video is all about electron microscopes. Now, electron microscopes are way better than light microscopes, and because of that, they cost about £1 million. Like, no joke, literally about £1 million. Now, here's why. So as we've talked about, magnification is an important property of microscopes, but if you continually increase the magnification of an image beyond a certain point, it doesn't increase the detail you can see. If you think about a picture on your phone, if you zoom in and it loads, the quality doesn't always stay the same. The quality actually decreases. The ability of the microscope to see in detail is called its resolution. So, like your phone, whenever you zoom in more, okay, you can see things bigger, but it's not clear because you're losing the resolution as you zoom in. The best light microscopes can resolve details that are about 0.2 micrometers apart and require a magnification of times 1,500 so that our eyes can see this detail. But electron microscopes use a beam of electrons to pass through a specimen, and they can see the details of the structures inside the cells. They have a greater resolution, being able to show details that are about 10,000 micrometers apart. The highest powered electron microscopes can even show detail of large molecules such as proteins, and they can have magnifications of up to 5 million. Now, this is what an electron microscope looks like. It looks very big, it looks very expensive, and it's taken up this whole area. So what they do, which is slightly different, instead of passing light through the sample, they actually shine electrons through them. So the electrons can either go through them or they can bounce off them. And those are two slightly different types of electron microscopes, but they give you loads of really cool images. So for example, here we can see a red blood cell and a white blood cell. And you can see here that the little scale bar's there. And that means that in real life, this red blood cell is about 5 micrometers in width. What you can then do with them is you can use computer programs to recolor them. So this is not real life. This was that image, but it's been recolored so that you can see different objects more clearly. With the electron microscope, you can also see inside the cells in far more detail. All the images we've seen so far of an animal cell have been done with light microscopes, where we've only seen the cell membrane, the cytoplasm, the nucleus. What you can actually see here is all this other stuff. This is all the extra detail. So this is the nucleus. This is a little thing inside the nucleus called the nucleolus. And then we've got all this other stuff. Here you can see loads of mitochondria. You can still make out the cell membrane and the cytoplasm, but you can see that there's actually a whole lot more going on. Here's a plant cell where you can see the vacuole, which is all this big clear area in the center. Chloroplasts around the outside. You've got the cell wall round here, and then you can see the cytoplasm there. So you can see in a lot more detail. Now, here's the advantages and disadvantages of electron microscopes. So the advantages are you can see in higher resolution, you can see in a higher magnification, can't you, Jenny?
Yeah.
The disadvantages are that they're large, expensive, you need special staff, and it kills the tissue that you're looking at.